Okay, great. So good morning, everyone. My name is Daniele Agostinelli, and today I'm going to talk to you about mutations in plant shoots, which is the topic of my PhD studies at CISA. And now it's a bit different from the research project I'm involved in at UBC. Anyway, let me thank the organizer for this nice event, Frontiers in Biophysics, and also the sponsors. And let me thank also my PhD supervisors, Professors De Simone and Noselli, and my collaborator, Dr. Luca Antonio. So we often think of plants as objects which do not move. And our misconception is essentially coming from the fact that we often focus our attention on very fast events. So we really need time-lapse imaging techniques in order to bring these slow motions to a time scale we are used to. In this way, we are able to detect and to observe an extraordinary variety of movements exhibited by plants while growing. So in these videos, you can see some examples of circumnutating plant shoots. They are exhibiting these three-dimensional trajectories, and if you project these trajectories on the horizontal plane, you will get some pendular, elliptical, or circular patterns. The literature on the topic is really broad, and the consensus is that these circumnutations derive from differential growth. So essentially, there is one side on the stem in which cells are growing more than the cells on the opposite side, and this generates a spontaneous curvature. However, the spontaneous curvature is in principle different from the visible curvature. Indeed, the visible curvature will depend also on external loadings. Despite the fact that this kind of movements have been observed long time ago, there is no consensus on their nature. So a long-lasting debate produced three main theories. The first theory is usually attributed to Charles Darwin, and it is the one of the endogenous oscillator. Essentially, according to this theory, these kind of oscillations are the result of prescription, endogenous prescriptions. So basically, if this is the cross-section of the stem, and this is the vector which gives you the direction of the differential growth, according to the theory, this vector is rotating across the cross-section following a biological rhythm. Then there is the second theory, which is the exogenous oscillator, according to which this is traced back to a paper in Gradman in 1922, who described these kind of oscillations as a spontaneous reaction of the plant to external stimuli. For instance, it is well known that plants react to gravity. So if you rotate a plant, the support of a plant, this triggers a correcting movement which tries to align the main axis of the plant towards the vertical line. So if a plant is deviating from the vertical line, this triggers a correcting movement, and due to some delay in the biological process, the plant is going to overshoot its target. And at this point, there is a reaction movement in the opposite way, which generates this kind of self-sustained spontaneous oscillations. More recently, there has been another theory, which is the two-oscillator model. So according to the theory, both endogenous and exogenous oscillations coexist in this phenomenon, and there are experiments on space which are supporting this hypothesis. Indeed, it has been proved that under microgravity conditions, gravity, if there is no gravity, these kind of oscillations are still present, but their amplitude is much smaller. So this new theory is very interesting, but leaves open some questions. So if it is true that there are both endogenous and exogenous oscillations, which one of them dominates, when, and why? So in order to answer these kind of questions, we developed a mathematical model based on the theory of morphoelastic rods. So we have a rod, which is a reference configuration, and then a current configuration. As usual in rods theory, we have these configurations, which are described by three directors, a tangent vector and a base, which defines the cross-section of the rod. And these directors, by means of this differential equation, define implicitly this vector field of strains. We assume that this vector field can be decomposed in an additive fashion. In other terms, there is one part, which is the elastic strain, which is due to a uniform distributed loading due to gravity. And then there is an intrinsic strain, which is visible when there are no external forces. If you have both intrinsic and elastic strains, then the visible strains will be the result of the two. It will be a sum. So at this point, we can write the evolution laws for the intrinsic strains by taking into account the plant reactions to external stimuli, such as gravity. If we do so, we realize that actually the straight position will undergo a mechanical instability. So this is the stability diagram on the parameter in the parameter space with on the x-axis you have the length of the shoot, and tau g is actually the time scale for the evolution of the spontaneous strains. So here we notice that for any given value of tau g, you have a critical value, a star, above which the plant is going to be unstable. So in the nonlinear regime, we observe the emergence of some periodic non-trivial solutions, which is an Hopf-like bifurcation, essentially. So we observe pendular oscillations or circular oscillations according to the initial perturbation that is provided. And these kind of oscillations remind us about circumambulations of plants. So now if we consider also an endogenous oscillator, we get epitrochoid-like patterns and hypotrochoid-like patterns for supercritical length of the shoot. So at this point, if we consider a shoot which is growing from an initial length which is subcritical to a final length which is supercritical, we see the transition of the shoot passing from an initial regime in which endogenous oscillations dominate the dynamics to an intermediate regime in which both endogenous oscillations and exogenous oscillations due to the Hopf bifurcation compete, to arrive finally to a stage in which exogenous oscillations are dominant. As a proof of concept, we conducted some experiments on plants, and here we report the trajectories for a qualitative comparison between the simulation on the left and the experiment on the right. So talking a bit more about the experiments, we grew some specimens of Arabidopsis thaliana at our lab. Here on the top left, you see the stereopero images. And we were able to reconstruct the three-dimensional trajectory here on the right. It seems that my video is lagging. I hope you can see well what's going on. Anyway, here on the left bottom, you see that there is the projection on the horizontal plane of this three-dimensional trajectory. And now it's very interesting to notice that also in this experiment, we observe a transition from a very first regime in which oscillations are very small and a later regime in which oscillations grow in amplitude. So in some sense, we are able, by means of this Hopf-like bifurcation, to explain why we see this kind of transition between one regime to the other one. And this provides at least a theoretical explanation of this phenomenon. So now I would like to stop here to give space for questions if there are any.